Hi, morning. I'm here to talk about the electron microscopy, its introduction, and some of its applications in sustainability development. Contents of my talk will be electron microscope, its two types, and its applications in sustainable development, and just some of it, a summary, and a quick tour about IEM. Before we start, we'd like to talk about the resolving power of a microscope in our eye. The smallest object of our eye can identify is about zero point 0.1 to 0.68 millimeters. But if you want to go beyond that, let's say, for example, if you want to see the thickness of a human hair, size of a blood cells, size of a bacterium, depending on the amount of microscope, we use an optical light microscope. If we want to see more details, we go to electron microscope, just like with the picture of the tardigrade. The electron microscope is very powerful. It's a high resolution that can see structures of crystals, and it can go to imaging the lattice structures of this material. Analyzing an object requires a certain form of stimulation. This stimulation can be a form of electromagnetic radiation. This is visible light. Okay, it can be an optical, can be UV, IR, coherent sources like lasers, and even X-rays in some applications. If you want to check, do an X-ray microscopy, and finally, electrons. So what is an electron microscope? An electron microscope uses an electrons and they stimulate the object in order to create an image. So the light photons substituted with electrons, and the lenses, which is used in glass lenses, are substituted with electromagnetic coils or electromagnetic lens to focus the electron beam. There's a similarity with an optical microscope and an electron microscope. Uh, so just to compare, the radiation sources is a visible light, while as electron microscope is an electron beam. The image, this is black and white for electron microscope, it's called and color. Best resolution for a light microscope is about 2,000 nanometers. An electron microscope can go beyond less than 1 nanometer and more than 200,000 x magnification. The medium of travel is air for light, and the electron microscope is vacuum. So this is the reason why it's not possible to image live specimen in electron microscope. It has to be completely dry. The lenses uses optical lenses, which is adjusted mechanically by an optical microscope. Whereas for electron microscope uses electromagnetic lens. So this is just a composed of coils, okay, where we adjust the current in order to modify the magnetic field. Electron microscope began in the discovery of electrons by J.J. Thomson. Okay? And de Broglie has a large participation in this because of the particle wave duality of the electron. It was only in 1927 that the first development of coils, okay, to focus the electron beam was done by Hans Busch. And the first truly electron microscope was built in 1931. It was only in 1960 that the first commercial type of electron microscope, like the scanning electron microscope, was marketed by Cambridge Instruments. So why electron? Electron has a very good magnification, resolution, and particularly depth of field. You can see the image on the right. A microscope has a very short depth of field. It can only shows you the surface. Whereas for an electron microscope, it can show you the background as well as the entire structure of this object. And why is electron microscope? Because of the high resolution, okay? and, and the wavelength is very small. Typically, for a 5 kilovolt scanning electron microscope, okay, can have a resolution of 0.212 nanometers. And the electron wavelength is around 0.173 nanometers, or about 0.1 angstroms. Compared it with the wavelength of visible light, this is way shorter compared to them. So you have a higher resolving power compared to optical microscope. Images are formed in an electron microscope due to its electron and matter interaction. Electron gun, okay, which is, produces the electron beam focused on the object, and you have a lot of interactions that produces different signals. These are secondary electrons, or SE, X-rays, backscattered electron, Uga electrons, and then for a very thin specimen, the transmitted electrons are either elastically or inelastically scattered, and some of them transmitted, and some of them are absorbed on the specimen. Okay, so these various signals, okay, can be used to construct images depending on the concept. Transmitted electrons are usually made for transmission electron microscopes, and these interactions on the surface are usually signals are usually used for scanning electron microscopy. So as I mentioned before, these interactions depends on the types of microscope. Okay. Surface interactions uses the scanning electron microscope, and transmission electrons microscope uses the transmission interaction. First, we'll discuss about the scanning electron microscope. Its basic components is just similar to an optical microscope, where you have the source, the lens, and the specimen. Okay, this source which passes through the a series of condenser lenses and projector lens onto an object. So the scattered electrons from the surface produces the signal. This. There are two types of signals, which I mentioned before in the electron-matter interaction. First is secondary electron imaging, or SEI. This is due to the inelastic scattering of a primary electron. This is emitted on the top of the interaction volume, the surface. Okay, it's about 1 to 15 nanometers depth. And this is mostly used for imaging surface topography. The other one is the backscatter electron imaging, which is about 300 nanometers from the interaction volume. So this is very slightly deep. And this is due to the elastic scattering of the electrons due to the nucleus. Consequences of this is that higher atomic number provides higher reflectivity and a brighter contrast. So let's see. Comparison of the backscatter and secondary electron images. You can see from the backscatter images, there's a material contrast. Okay, so materials with a higher atomic number, okay, has a higher reflectivity because of the scattering, and then the materials with the low atomic numbers has a less scattering, so this appears darker. Whereas for the secondary image, you can see only the surface features of the material, but not any material contrast. The second image shows the same thing. Okay. This is a material contrast. Okay. This is a higher atomic number. Whereas for the secondary image, you cannot see anymore this one, but you can see the surface features where there's a lance and roughness shown on the surface. So secondary images are useful for if you want to know the structure and this roughness of your object. And the backscattering images is very useful if you want to see material contrast or difference in material in your sample. Acceleration voltage plays a good part in electron microscopy. 5 kilovolt, it's usually around about 5,000 volts. And then this is 10,000 volts. You can see the difference. Okay, this is for the secondary image. Whereas for this one, 3 kilovolts, you can see the surface features of this sample. Whereas if you move up to 20,000 volts, the details of the surface is gone, because acceleration voltage means higher penetration on the surface, and then you see less details on the surface. optics. One good thing about this scanning electron microscope or electron microscope in general is its ability to do quick elemental analysis using energy dispersive X-ray spectrophotometer. It allows a quick material elemental identification. It's a quantitative and qualitative techniques which gives elemental composition of material with atomic numbers greater than 3. So how does it work? Okay, so the primary beam, as I said before, for electron matter interaction, okay, when an electron hits an electron from in this shell, okay, it is being dislodged, and then another electron from K from another shell in L, okay, occupies vacant this vacant hole, and then this relaxation results into characteristic X-rays, which shows and process shows the elements of this material. So that's how EDS or EDX works. Okay, it's just the electron beam and then the X-ray, and then it's being uh, collected by this detector, silicon dip detector, and then it's processed to produce this spectrum. So from the spectrum, the software automatically gives you a distribution of different elements, okay, from a mass percentage to atomic percentage. Of course, it can give you a qualitative analysis like elemental mapping. The scanning electron microscope, this sample can see this distribution of this manganese, aluminum, as well as iron. This EDX instrument is usually located at the side of an electron microscope, which is a protruding. It's just a special option actually. In some cases, it is included. In some cases, it's not. EDX works by higher acceleration voltage. There is a such thing as overvoltage ratio. It's a ratio of acceleration voltage versus the ionization energy. The ratio ideally should be 2 or greater than, or more or less 2. Too much high over voltage will degrade the spatial resolution. So just an example, a specimen being irradiated with a 220 kilovolt electron, okay, we show more elements compared to a 5 kilovolt, okay, uh, acceleration voltage. Now we're going to a transmission electron microscope. Transmission electron microscope is basically similar to a scanning electron microscope, but this one, it uses the transmitted electrons and then processed to an imaging plate or camera below the sample. It is a very tall instrument. It's about 2.5 to 3 meters in height depending on the acceleration voltage. So you can see from the right, just an example, a 3 million volt transmission electron microscope compared to a researcher. So you just imagine how high this instrument is. One. Okay. So this is composed of electron source, electromagnetic lens system, the focus and shape the beam, sample holder, which is in the middle, and then you have the projection lens, and then onto the CMOS camera. Okay. So this is the difference. The sample in a scanning electron microscope is below, below. This is the end point. But in a transmission microscope, the sample is injected in the middle of the column. So working principle of a TEM is that we have illumination, the lens are focused on the sample, okay, and then we have the objective lens and projector lens onto the sample. So that's where transmission, okay. Electron beams are transmitted on the sample, but it has to be electron transparent. It has to be less than 100 nanometers. Solid state materials does this by using focused ion beam in order to develop this kind of lamella. For biological samples, there's a special method called ultramicrotome to create these very thin sheets of materials, okay, in order to be electron transparent. These samples, okay, these very thin samples are placed in the copper grid, which is this grids with carbon film and foam bar in order to place a sample onto this um, grid spaces. Okay, this grid is about 3 millimeters in diameter, and then this is placed into a holder, okay, locked into place, and this holder is inserted into the middle of the column. There are three components of imaging formation in a transmission electron microscope. Is absorption, okay, for low density, okay, can pass through. For high density, okay, some of it are being absorbed and then can cause damage. Then scattering for low density, it can pass through. Some are less scattered, and higher density, more scattering, okay, and shows in the objective aperture. And then finally, diffraction and phase contrast, okay, uh, because of the objective disc and projector lenses, okay, the beam are being shaped, okay, in accordance with the density of the material. So these three types of absorption, scattering, diffraction phases are actually a convolution of the are actually a summation. Okay, so image are formed from these uh, uh, three processes. This is an example of a TEM image. So this is a TEM image of a barium nitride using bright field and dark field. And like scanning electron microscope, it can be utilized to do elemental mapping as well. And because of the resolving power of transmission electron microscope is much higher compared to scanning electron microscope, can also, okay, image the lattice structure as well as the electron diffraction pattern of this material. Aside from that, of course, it's very useful or widely used in biological studies, like for example, in this mouse killing. As a comparison, TEM and SEM, transmission electron microscope has a higher resolution. Okay. It can resolve up to 0.05 to zero point 0.3 nanometers, whereas the magnification can be from 1,000 to 2 million x magnification, and acceleration voltage is 40 to 300,000 volts. So that means it's a much shorter wavelength compared to scanning electron microscope. There's a lot of images that can be derived from TEM, which is the TEM image, STEM, or scanning transmission electron microscope. We can produce a bright field and dark field, electron diffraction images. Whereas for scanning electron microscope, we can only have secondary electron and backscatter as a basic unit. In some cases, there are special detectors that uses deuterium aluminum garnet or YAG ESE detection system. Both can do EDX, and the sample preparation is very complex for TEM. It requires very thin sectioned materials or ultra microtome for biological samples. For SEM, it's a simple conductive base, carbon tape, and then sputter coated to avoid charging. And this TEM is a very tall instrument, requires a ceiling required at more than 3 meters. You have to have a room which is a high ceiling and a three-phase power system for its cooling, because you have to cool down the electromagnetic lens, which uses a lot of current. Right now, SEM is there's a lot of models. SEM, even bench type, this is very compact, so that much room requirement, because it can fit to in a small room. And of course, the cost of a transmission electron microscope is exorbitantly high compared to scanning electron microscope. Now we'll give you some application sustainable development. So there are about 70 of them, especially targets and the sustainable goals in the Philippines. But I'll just get a few of them from this talk. First is the development of renewable energy sources. So this is one of my most favorite topic is the triboelectric nanogenerators. Okay, so triboelectric nanogenerators are sensors that you can be attached to your body in such a way your movement can cause contact separation on these sensors, thereby producing electricity. This is what we call triboelectric generators. Okay? So scanning electron microscope is very useful in analyzing the surface, how rough it is, how smooth it is, in order to generate sufficient triboelectric energy. And of course, for climate action and good well-health being, the scanning electron microscope is useful in analyzing haze. Like for example, this paper from Journal of Environmental Protection that they analyzed the haze composition of in Xi'an City in China. So I think it's very useful also. It's very interesting. We would like to know the haze composition of Taal volcano in the surrounding cities. And of course, it's also useful in studying the microplastics, contaminants, and as reported before, these microplastics are even found in human tissues, which is uh, very bad for our health. And of course, soil analysis can be used for soil analysis, like this fly ash incorporated biocement to prove the strength of expansive soil. It can magnify the soil components in such a way to know if it is properly mixed or not. Okay, and then you can have elemental analysis to see the difference when these components are added. For TEM, TEM is very useful for the field of botany and biology. So this is an example of an image of a fragrant rice to improve crop cultivation, as well as the wheat grain. And also for marine studies, TEM is also useful in analyzing the growth of corals. And it's interesting that for coral, you can also analyze the crystal structure lattices as well as the electron diffraction. So as a summary, electron microscope is a good instrument to use electrons to excite the material in order to generate high voltage resolution. Its magnification is many order higher and superior depth of field compared to optical microscope. However, it has a few disadvantages such as it needs high vacuum to operate and not possible to view live specimens. The electron microscope, a versatile instrument, has found its way in sustainable development research, which I gave some few examples. So lastly, before I finish this talk, I just want to give you a tour of our integrated electron microscopy laboratory. So we have the scanning electron microscope and the transmission electron microscope. So we have here the 200 kilowatt TEM. This is one of the most powerful electron microscope in the country. So we can do bright field, dark field, EDS, as well as diffraction imaging. And here, this is a semi-clean room environment. Okay? It has to pass through the air shower. And we have here the focus ion beam for preparing lamella samples as well as do micro cross-sectioning. And to the left is the scanning electron microscope. We have also here for my micro manipulator, a clean bench lab with a wrist strap for handling electrostatic sensitive devices, as well as the spotter coater. And we have a separate room for preparing biological samples, which contains the ultramicrotome, some ovens, centrifuges, and some rotators for preparing resins in embedding the biological samples. So thank you very much. Okay, thank you.